Hello students, we are learning autotrophic nutrition. We have seen that in autotrophic nutrition, the photosynthesis process occurs in which carbon dioxide and water reacts with each other and making glucose and oxygen as a byproduct in chlorophyll and sunlight. In this photosynthesis process, carbon dioxide, sunlight and chlorophyll are the most necessary materials to carry out the photosynthesis process. Now, how do we prove it that chlorophyll, sunlight and carbon dioxide is necessary? Let us see the activity here. Our aim of this activity is we need to prove chlorophyll is necessary for photosynthesis process. For that, we are taking the apparatus, a potted plant with variegated leaves. What is variegated plants? These are the plants which is having a which is having the leaves of different shades. It means the green shade having the chlorophyll pigment in it, and a different color will be having a different colored pigment in the leaves. So the green area will represent the chlorophyll pigment and other shades having will represent a different pigment. So a plant having a variegated leaves, beaker, sheet of paper, alcohol, water bath, Bunsen burner, dilute solution of iodine. The iodine solution will be used to see the presence of starch. Now let us see the procedure. So if chlorophyll will not be present in the leaves, the starch means the photosynthesis process will not occur, and or we can say that the area of the leaf having a chlorophyll, on that area only the photosynthesis process will occur. So we have taken a variegated plant. First process we are doing the destarching process. So this process, what is this process? See, the plant are doing the photosynthesis process every day. So in the leaves of the plant, the photosynthesis process has occurred. So starch will be produced. In the photosynthesis process, first of all, glucose is produced. This glucose that is having the chemical formula of glucose is C6H12O6. It means a molecule having 6 carbon. So when this glucose is formed, it goes through three different ways. So first one, the glucose will be used in the energy production, which is used by the plants. Second one, this glucose will be used to make cellulose, protein in the plants and different materials. And third way, the molecules will join a long chain of carbon, not the six number of carbon, but the six number of the molecules having 6 numbers will be joined together and making a long chain of carbon. That carbohydrate we are calling it starch. So starch is nothing but a long chain of carbon. So here, when the photosynthesis, the earlier photosynthesis process has made the starch in the plants. So in the leaves, the starch is already present. So when we directly do the starch iodine solution test, it will give a starch, presence of starch, which is already occurred in the previous test. So first we need to remove this starch which is already present from the previous photosynthesis process. So for the destarching process, that means to remove the starch which is present in the leaves by the previous photosynthesis process. So for that we are keeping this pot into a dark room for 2 to 3 days. So in this 2 to 3 days, in the dark room, there will not be any sunlight. So photosynthesis process will not occur. So starch production will not be there. And the excess starch will be used up by the plant. So in 2 to 3 days, the earlier starch will be used up. Now if we want to do the process, we are keeping it into sunlight for 6 hours. In this 6 hours, photosynthesis process occurs and the new starch will be produced. New glucose will produce, and that glucose will ultimately convert into starch. Now, to check the presence of starch, take a leaf of that, take a leaf from the plant, mark a green area. Okay, it is a variegated leaf, so it has some area as green, which represents the presence of chlorophyll, and some areas having different pigments that chlorophyll will not be present. The next step is dip the leaf in boiling water. When the leaf is dipped in the boiling water, the cell membrane of the cell will be ruptured, so the cell will die, and as the cell membrane is ruptured, it is broken down so that we can test the starch present in the cell very clearly. So the leaf is kept in a boiling water, then immersed it in the alcohol. Place the alcohol beaker in water. Water the alcohol boil, and then we are keeping the leaf in a alcohol, and this alcohol beaker will be kept in a water bath. So when the water will, and we heat this water bath, so water will boil and ultimately the alcohol will boil. We are not directly boiling the alcohol by on keeping in the burner, because when we burn the alcohol, it is a volatile substance, so it will burn, right? So we are keeping this alcohol into water bath, and as the water will boil, inside alcohol will also boil. So why we are boiling this leaf into alcohol? Because when we boil this leaf into alcohol, the chlorophyll will be absorbed by the alcohol. Okay. So now there will not be chlorophyll inside the leaf, and the leaf will become completely pale. The green color of the leaf will be removed because now chlorophyll is absorbed by the into the alcohol. That's why, because in why we are doing this, because if chlorophyll will not be present, there will be a clear color change into the iodine test. So to take a clear iodine test, to make the accurate iodine test, the leaf is kept into the alcohol, and alcohol will absorb the chlorophyll. So next step is the color of the leaf changes from green to colorless as we are boiling into the alcohol, and this dip this leaf into iodine solution. And by keeping into iodine solution, wherever the starch will present, see, iodine solution is used to check the presence of starch. So if the photosynthesis process has occurred, the starch will be produced, and if the starch is there, it will change the color of alcohol solution. Alcohol solution is a purple color solution, and when it reacts with starch, it will give the blackish brown color. So wherever the starch will be present on the leaf, what we are observing, see, these are the areas, let us assume that these are the areas having chlorophyll. Now if we are keeping into iodine solution, the areas having chlorophyll made the starch by the photosynthesis process, and as the starch is present in that area, it will give a blackish brown color of the iodine solution on the leaf. It means that wherever there is chlorophyll, the photosynthesis process can occur, and the area where chlorophyll is not present, there will not be any color change, which means that chlorophyll is necessary for the photosynthesis process. So this was our activity to prove that chlorophyll is necessary for the photosynthesis process. Next we are trying to prove that carbon dioxide is necessary for the photosynthesis process. So our aim for the activity is carbon dioxide is necessary for the starch formation, that is for the photosynthesis process. For that, our apparatus are two healthy potted plants of same size. Any plant we can take, two bell-shaped glass jars, watch glass, KOH, potassium hydroxide, Vaseline, and glass plates. In this activity, the procedure is as follows. The first step is of our experiment is destarching process. It means that we have seen already that destarching means to remove the starch which is already present in the leaf by the previous photosynthesis process. So in this we are keeping both the plants into a dark room for 2 to 3 days. Okay, so it will use up all the starch formed inside the leaf. Now next, place both the plants into glass plates and cover the plants into by the bell-shaped jars, glass jars. And in one of the jars, we are keeping a watch glass having potassium hydroxide in it. And then seal the bottom with the Vaseline so that we are keeping it completely airtight. Place watch glass containing KOH in one of the jars. Okay, so in watch glass we have potassium hydroxide in one of the glass jars. Keep both the plants in sunlight. Now, for the photosynthesis process, we are keeping this both plant into sunlight for 2 to 3 hours. So, photosynthesis process will occur. Keep now the next step, pluck a leaf from both the plants. After that, the photosynthesis process has occurred. After that, we are taking leaf from both the plants and check the presence of starch with the iodine solution. So, if photosynthesis process has occurred, it will form glucose and glucose will ultimately form starch. So, if we are checking the presence of starch with iodine solution, if it change the color of the leaf, the starch is there. And if color change is not there, starch will not be there. So what do we observe by in both the leaves? So we are observing that the leaf of the pot with KOH, that is figure A, means having potassium hydroxide in the jar, will not turn blue black, and the leaf of the pot without KOH, that is figure B, will show blue black color of color. What does it mean? That the potassium hydroxide has the ability, is used here to absorb the carbon dioxide. So this in figure A, potassium hydroxide KOH will absorb all the carbon dioxide present inside the glass jar. So carbon dioxide will not be available for the plant for the photosynthesis. And in figure B, KOH is not present. So the carbon dioxide is available for the photosynthesis process. So this leaves will will do the photosynthesis process and form the starch. So the color, and when we are keeping it into iodine solution, the color will change to blue black. And this leaf without with KOH, photosynthesis process has not occurred. That's why it will not change any color. So by this activity, we can prove that carbon dioxide is necessary for the photosynthesis process. So these are the two main activities which shows that chlorophyll and carbon dioxide is necessary. And third, sunlight is also necessary. And how do we prove that? If we keep the plant into dark room, then or we can cover the one stem of the plant with any dark or opaque material, then that leaf or that plant will not going through the photosynthesis process, which means that sunlight is also necessary for the photosynthesis process. Students, read up to this. We are in next lecture, we are going to do the heterotrophic nutrition. If you have any doubt, ask me. Thank you.